Hello students, welcome to SANDEEP online biology classes. So today is the third day, almost two sessions we completed in the sexual reproduction flowering plant. Before we start this chapter, so ಎಲ್ಲರಿಗೂ ದೀಪಾವಳಿ ಹಬ್ಬದ ಶುಭಾಶಯಗಳು. ಸೊ ಎಲ್ಲರ ಜೀವನದಲ್ಲಿ ಬೆಳಕು ಬರಲಿ, ಅಂದರೆ ಜ್ಞಾನೋದಯ ಆಗಲಿ, ಎಲ್ಲರೂ ಆರಾಮಾಗಿರಿ, ಆರೋಗ್ಯವಾಗಿರಿ, ನಗ್ತಾ ಇರಿ, ಸಂತೋಷವಾಗಿರಿ ಮತ್ತು ಸಮಾಧಾನಿಯಾಗಿರಿ. Yes, so straightway we are going to the chapter, means the content we are not covered in the last two sessions. So almost the last year video NCERT book in content ಕೊಟ್ಟಿದ್ದಲ್ಲ ಅದೇ ರಿವೈಸ್ ಮಾಡಿದ್ದು, ಫಿಗರ್ ಬೇಸ್ಡ್ ರಿವಿಷನ್ ಇದೆ. ಸೊ now the leftover topics which are from the CET point of view and the Neet point of view is very important one. So now on the main point, so your topical ಆಗಿತ್ತು sexual reproduction flowering plant means the flower is the special parts or the organs of the plant which are specifically meant for the reproduction of it. The flower, the typical complete bisexual flower consists of four whorls: calyx, corolla, androecium, gynoecium. The calyx is made up of sepals, corolla is made up of petals, androecium is made up of stamens, gynoecium is made up of pistil. So these two words, calyx and corolla, are the accessory and these two are the necessary ones. Yes. So if it is very difficult to identify which is the calyx, which is the corolla, then the particular word is called the perianth, and that perianth is made up of tepals. Yes. So if the appendage of this calyx corolla is fused one, then gamo, the term word used. If they are free, then it is called the poly. Gamosepalous, gamopetalous, polysepalous, polypetalous. So these are the morphological points. Now the main one, in this chapter we are going to cover what is happening in this male reproductive part, that is androecium, and the female reproductive part, that is the gynoecium. Yes, only these two parts we are going to study. So first one is androecium, and gynoecium. So in chapter the heading is a pre-fertilization structure and event. Pre-fertilization means before the fertilization. So these structure we have to cover, and whatever the event take place in this structure we are going to cover. So under that one, the heading is stamen, microsporangium and pollen grain. Just visualize. Stamen means it is an individual part of the androecium. Yes. Microsporangium means it is present inside that anther. Pollen grain means it is a male gametophyte which is produced in the microsporangium, released from the anther. So all these structures are related to male reproductive part only. Yes. So first one is the stamen. So this is a typical stamen in which two parts are there: filament and anther. Yes. Filament is a long, slender, thread-like structure. So this part is attached to the thalamus. Sometimes it is attached to the petals also. That condition is called the epipetalous condition. At the tip of this filament, the anther is there. The anther usually, so elongated, the center of this one connective tissue is there. Yes, the type of anther again it is a different one. So basifix, dorsifix, verticillus, different types of anthers are there. So that is different. The anther, when you take the transverse section of this anther, if you cut this one, transverse here, then you will get the rough figure like this one. So to explain this one again, many terminologies there. So the transverse section of the anther, we show the almost tetragonal structure. So four lobes are there. So it is a tetragonal structure. So it is a diathecous bilobe tetrasporangiate structure. So diathecous means this is one theca, this is one theca. So bilobe means yes, it is a bilobe. This is one lobe, this is one lobe, and each lobe is consists of two theca, one and this one. Yes. So in each lobe, two microsporangiae there. That's why it is a collectively tetrasporangiate structure. Four microsporangiae. So this is the young anther. So how it developed? So how this formed means this structure is formed. So main one, when the anther is young one, yes, still it is not differentiated like this one. When it is a young anther, some of the hypodermal cell differentiate and they develop as a archisporial cell. Yes, archisporial cell. So these cells are little bit prominent and filled with a large amount of cytoplasm and prominent nucleus is there. This archisporial cell divides and produces two types of cell. This cell again divides, produces two cells, primary parietal cell and inner sporogenous cell. So this is the archisporial cell. This will divide and produce two cells. So this is a primary parietal cell and this is a primary sporogenous cell. Two cells are formed. So now further division is different one. The primary parietal cell undergo repeated division. So repeated division, and this will produce the anther wall, outer protective layer. Now primary sporogenous tissue which is present inside, this will also undergo further division, and this will produce the microspore mother cell to a sporogenous tissue theory. Yes. So even archisporial cell divide ಆಗಿ primary parietal cell ಆಯಿತು ಮತ್ತು primary sporogenous tissue ಆಯಿತು. Primary parietal cell ಮತ್ತೆ divide ಆಗಿ anther wall ಆಯಿತು, and this is produce the sporogenous tissue. This primary sporogenous cell produce the sporogenous tissue which is made up of microspore mother cell. When you take the anther wall nothing but these layers. Yes, the anther wall is made up of epidermis, endothelium, middle layer and tapetum. ಅಂದ್ರೆ ಈ anther wall ಮತ್ತೆ ಡಿವೈಡ್ ಆಗಿ epidermis, endothelium, middle layer, tapetum. All these layers are produced from this primary parietal cell. The question ಕೇಳಬಹುದು, the epidermis, endothelium, middle layer produced from which cell? Primary parietal cell. And sporogenous tissue produced from the primary sporogenous cell. Yes, so these are the mother cell. So gradual differentiation occurs and ultimately the anther will form like this structure. ಈ type structure develop ಆಗುತ್ತೆ. So ಈಗ ಸೆಂಟರಲ್ಲಿ ಕನೆಕ್ಟೆಡ್ ಟಿಶ್ಯೂ, ಇದೊಂದು ಲೋಬು, ಲೋಬ್ದಲ್ಲಿ ಎರಡು ಮೈಕ್ರೋಸ್ಪೊರೆಂಜ್ ಇದೆ. So now stamen structure is over. This is the stamen. ಈ microsporangia means only this structure we have to study. Only this one corner. In microsporangia ಅಂದ್ರೆ in ಒಂದೇ corner. Yes. So again the elaborate description is given in the NCERT textbook. Microsporangia means it is covered by the again four layers: epidermis, endothelium, middle layer, tapetum, inside sporogenous tissues. Yes. So ಇಲ್ಲಿ main point of view that how these layers will function. ಈಗ epidermis ಇಂದ ಹಿಡಿದು tapetum ವರೆಗೆ ಪ್ರತಿಯೊಂದು ಲೇಯರಿನ ಕೆಲಸ. So we will take these are, first one is the epidermis. So these are made up of barrel shaped cell, elongated cell. So only one layer. So this will give the protection. Endothelium is also elongated cell, almost 2 to 3 layers. Endothelium is there. Difference means this endothelium cell, this is the endothelium cell, it's a radial wall and tangential wall. So if you take the layer of cell, so this radial wall and tangential wall is thickened. So ಈ ಮಧ್ಯದಲ್ಲಿ ಗೋಡೆ ಮತ್ತು ಒಳಗಿನ ಗೋಡೆ, ಸೊ ಇದರಲ್ಲಿ ಸ್ಪೆಸಿಫಿಕ್ ಕೆಮಿಕಲ್ ಇಸ್ ಡಿಪಾಸಿಟೆಡ್. ಸೆಲ್ಯುಲೋಸ್ ಇರುತ್ತೆ, ಪೆಕ್ಟಿನ್ ಇರುತ್ತೆ, ಲಿಗ್ನಿನ್ ಇರುತ್ತೆ. So due to presence of this toughest material, so this endothelium layer is also called as fibrous layer. Yes, this will also give the protection, and collectively epidermis, endothelium ಇವು mature anther ನಲ್ಲೂ ಇರುತ್ತೆ. Yes, so main point, this epidermis and endothelium are very thin at this point. ಏನಿದೆ ಅಲ್ಲ, ಈ thick layer ಗಿಂತ. So this is the epidermis endothelium. This is also epidermis and endothelium. Yes, E point at the epidermis endothelium is the thinnest one. Yes, the point of E point. So it is a very thin point. So here deposition is very minimum. So the significance of this point will cover just one. Epidermis, endothecium is what? Next one is a middle layer. Middle layer is almost 3-4 layers. If you take the middle layer, so it is made up of 2-3 layers. Middle layer made up of 2-3 layers. So this green color, middle layer one. So this is present only in the young anther. Anther hang mature agutala. This middle layer degenerate, and from this only the nourishment is supplied to the developing microsporangium. So this will provide the nourishment. Mature anther alli middle layer irudilla. Means yavaga mature anther alli madhulu ella middle layer ella degenerate agi ee microsporangium say pollen sac anta onde chamber. So middle layer only. Epiderm is the most important. So in last class also in the first video, here epiderm function now will cover. So now try to cover the epiderm function. Epiderm functions is one day. Yes. First one, according to NCERT textbook, it provide nourishment for developing microspore. This is the first function. Yes. So in nourishment, yak month. So when you take the cells of the epiderm, if you take the cells of the epiderm, these are also elongated a little bit, but they contain dense cytoplasma and more than one nucleus. ಒಂದಕ್ಕಿಂತ ಹೆಚ್ಚು ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. So that is condition called a polynucleate condition. ಈ polynucleate condition ಹಿಂಗೆ ಜನರೇಟ್ ಆಗಿದೆ. So this condition due to nourishment ಎದಕ್ಕೆ ಕೊಡುತ್ತೆ? Due to polynucleate condition. So this polynucleate condition is happened due to the endomitosis. Endomitosis means karyokinesis occur but not followed by the cytokinesis. That's why the single cell contain more than one nucleus. So if the more than one nucleus are there, they will express in exaggerated manner, and this is helpful for the providing the nourishment for the developing microspores. So this is main. Next one. So after nourishment, this epiderm, second function, it produce Ubish body. Ubish granules. So this is main. So these granules are deposited on microspore and secretes the specific substance, secretes sporopollenin. Secrete the sporopollenin. So these Ubish bodies are deposited from the microspore, which are future pollen paper, and this Ubish granules secrete the sporopollenin. The sporopollenin is the toughest material which can withstand even the higher temperature, alkali condition and the acidic condition. Due to that one, these microspores can be easily preserved for a longer period in the nature. So sporopollenin is secreted from the tapetum. So functionally this is a very, very important one. Yes, Ubish granules and sporopollenin. Third function, it produces one more important substance, that is a pollen kit. Pollen kit. So this pollen kit is produced in the entomophilous flower. Entomophilous flower means insect pollinated flowers. Yes. So pollen kits are produced, which helps in attracting insects. Yes. So pollen kit. So three functions. Nourishment is one. This is the first function, only due to polynucleate condition. Ubish granules are produced. So this Ubish granules secrete the sporopollenin. This is toughest material. The sporopollenin is present in exime layer. Yes, the exime pattern will be decided by the sporopollenin only. Now next one, it produces the pollen kit that is help in attracting the insect in the entomophilous flower. This pollen kit also gives the protection against the UV radiations also. Yes, so this is it. One more thing, this pollen kit is produced by the specific tapetum. Still I am not known. Tapetum olage ಎರಡು ಇರುತ್ತೆ. So this pollen kit is produced by only glandular tapetum. Yes, each tapetum is the same producer. The pollen kits are produced from the glandular tapetum. Next one function, it produces one more enzyme. Fourth one, it produces enzyme called as calase enzyme. Calase enzyme. So, calase enzyme here help on. So, in last class you heard that the microspore in subplay, all microspores on attach tetrad root. So, this is a microspore tetrad. So, this microspore tetrad is covered with the specific chemical that is called the callose. Yes, it hold money. So, to dissolve this one, this calase enzyme will come and this will dissolve this calase, and due to that one, so callose dissolve ಮಾಡಿದ್ರೆ ಈ microspore separate out. So 4 functions are very very important. Apart from this tapetum will produce the enzyme like indolestic acid. Yes, some hormones are produced. So tapetum function is very very important. It is very easy to remember. Nourishment one to ಎರಡನೇದು. So nourishment ಎದಕ್ಕಾಗುತ್ತೆ ಹೇಳಿದ್ರೆ that is a polynucleate condition. ಎರಡನೇದು, it produces the Ubish granules that will secrete the sporopollenin, which is the toughest material, which will decide the exime patterning also. Yes. Next one is the secrete the pollen kit from the glandular tapetum body. So this will produce in the entomophilous of the insect pollinated flower. Next one is the calase enzyme that will help in the dissolution of the callose material from the microspore tetrad. So tapetum function is over. Now in tapetum only again types are there. Just cover that one. So here only I am going to write. Yes. Okay. So tapetum so differences in that one, 2 types are there. One is amoeboid tapetum. Second one is glandular. Amoeboid tapetum also called as a invasive tapetum. Glandular tapetum also called as a secretory tapetum. Yes, difference something there. So this is the sporogenesis tissue. This is also sporogenesis tissue. Yes. So surrounding that one, the tapetum is there. So a tapetum on imagine this is a tapetum and this is also tapetum. Now in this case, amoeboid tapetum or invasive tapetum means, so this is a tapetum. In this one, the radial wall between this one, again either dissolve either almost one on the other cell merge. And this will produce the pseudopodia, means the projection inside the sporogenous tissue. In projection so that's why it is an invasive. So it will penetrate inside the sporogenous tissue and provide the nourishment. Yes. So means after that one, it also degenerates. Anther mature ಆದ ಈ tapetum degenerate ಆಗುತ್ತೆ. But in this case, initially this glandular tapetum is intact. You dissolve intact and that secretory means whatever the nourishment is there, they will secrete inside. Yes, they will not produce the projection. So roughly tapitums are two type: amoeboid tapetum or invasive tapetum, glandular tapetum or secretory tapetum. When you take this pollen kit, is produced from this glandular tapetum, not from the amoeboid tapetum. So this is the main point. So just go through the tapetum function properly. This is an important point from the exam point. Next one is the microspores. How the microspores are produced inside this microsporangia. Gradually, this microsporangia develop into pollen sac. So what happens? So these are the now monitor. The archisporal cell divided into primary parietal cell and primary sporogenous tissue. Primary sporogenous tissue develop into microspore mother cell or the pollen mother cell. This is a diploid in nature. So here ploidy is important one. So primary pollen mother cell or microspore mother cell, it is a diploid. Yes, this undergo meiosis and produce microspore tetrad. So all are haploid in nature. 4 microspores are produced and all microspores are held together by the common bond that is called the callose. Yes, the suttalo callose irutte, the madhyadallu callose irutte, all are held together. So that's why separation is impossible. But this callase enzyme will come and dissolve this one and all microspores separates. And each microspore have the potential to develop into pollen grain. So these are the pollen grain. Yes, means if you observe this one, if you take any higher organism, so pollen grains are nothing but the male gametophyte, where the male gametes are there inside. So ultimately, from each microspore in there means thousands of microspores of the pollen grains are produced. Every microspore will develop into pollen grain only. Now, the microspore tetrad may be different types one. Now, based on the alignment of this one, microspore one. So, microspore tetrads are either this one or they arrange like this, otherwise this type, or step 2, otherwise one already I drawn, or this type. The type of microspore tetrad is also important. So these are the different types of microspore tetrad. So tetrad means, so this is a tetrahedral. So 4 corners are there, for the 4 corners 4 microspores are there. This is isobilateral. This is the isobilateral. This is decusate type. So if 4 are there, one is on that side. These are the 4 which are present on the lateral side only. So here 2 are present in this manner, 2 are in this manner. So orientation is different. This is the linear one, and this is T shape. Yes. So these are the different types of microspore tetrad. Now after microsporogenesis, the microspore develop into pollen grain. The maturation of the microspore takes place. So during which, so here when you take the microspore initially, so it has only one nucleus. If you take this microspore, it has only one nucleus. So gradually on this one, the different chemicals are deposited, and this will undergo maturation and gradually develop into pollen grain. So this is again same microspore, 2 layers are there. This is the intine, this is the exine. Intine and exine. Now, the nucleus which is present in the young microspore divide unequally, means here equal division will not occur. So nucleus division occur, but two different shaped cells are produced. The larger cell is called the vegetative cell, smaller cell is called the generative cell. Initially this generative cell is attached towards the one periphery. After that it will float in a cytoplasm of this generative cell. Now here comes the two layers. The polar grain wall is made up of intine and exine. Intine is made up of pectin and cellulose. Just recall. Exine is made up of sporopollen. Yes. So exine is made up of sporopollen, which is secreted from the tapetum only. The exine. So more topic is there. So I am going to write here. That topic covered. This is the polar grain. Yes. So this is the nucleus of generative cell and vegetative cell, and this is the intine, which is elastic and flexible in nature, which is made up of pectin and cellulose. Outside, sporopollen is deposited. The patterning of this sporopollen is specific for specific species. In some spaces, the sporopollen is not deposited. Yes. So this is about the pollen grain, and this is the generative cell where the two male gametes are there. So now this one, exine. The exine is made up of this sporopollen. Intine is made up of pectin and cellulose. The exine is again divided into two layers. So outer is exine, inner is end exine. End exine means equal to inner exine, ect exine means outer exine. This ect exine again divided into three layers. So ect exine again divided into three layers. So this is a foot layer. Yes. So these extension pillar-like structures are the baculate, and this is the tectum. So ect exine again divided into three one. So this is foot layer. This is baculate. This is tectum. Yes, the tectum pattern is variable. Sometimes it is a sharp, sometimes it is a blunt one. So tectum pattern is a deciding factor. So foot layer is a basic, it is a continuous one. Baculate is a discontinuous, it is made up of pillar-like structure, means in between that one space is present. And tectum is also continuous here. So tectum pattern is a main layer. So here the exine is again two layer, end exine, ect exine, foot layer, baculate, tectum is comes under the ect exine. So this is about the main one. So wherever the germ pore is not deposited, means sporopollen is not deposited, that is called the germ pore. The germ pore is there. So exine is important. This is the germ pore. So germ pore may be in spherical manner, E-type open space ಅಥವಾ longitudinal furrow ಆಗಿ ಹೀಗೂ ಇರುವುದು. So elongated furrow here. So germ pores are either circular or furrow type one. The base of the germ pore, if germ pore yes ಇರುತ್ತೆ. So if the germ pore 1, 2, 3 ಇತ್ತಂತಿದೆ. So if the only one germ pore is there, that is a monocolpate. ಎರಡು ಇತ್ತಂತ ಹೇಳಿದ್ರೆ bicolpate. ಮೂರು ಇತ್ತಂತ ಹೇಳಿದ್ರೆ tricolpate. So monocolpate usually seen in the monopods. Yes. So ಒಂದೇ germ pore ಅಲ್ಲಿಂದಾನೆ pollen tube ಹೊರಗೆ monopod. So bicornate tricornate mostly present in the dicots. Yes. So monocornate, and again if the number of germ pores are more, the angle between the germ pore is a deciding factor. Every species has a particular arrangement of the germ pore. So taxonomically, the germ pore no identify ಮಾಡಿ ಭಾಳ decide. So this is about the male reproductive part. And one more thing, the study of the pollen grain, again microspore develop ಆಗಿ pollen grain ಅಂತಲ್ಲ, the study of the pollen grain is called as a paleontology. So when you take the honey, in that also pollen grains are there. So this is about the male reproductive part. So where some neat points will be covered. So after a short pause, again we are going to cover the female reproductive part, where the anatropous ovule and the types of embryo sac we are going to cover. So this is about the stamen, microsporangium and pollen grain. So we will continue this class after a short pause. Yes students, now we are going to cover the female reproductive part. So only few topics are there, just revise in the fast one. So pre-fertilization structure, we have carpel, megasporangium and embryo sac headings. So carpel is a single unit of the gynoecium. Megasporangium means it is nothing but the ovule. Yes. So embryo sac means it is a female. Can lead to 5. Same when you take the male one, stamen they are given, microsporangium is given, inside the microsporangium, pollen grains are produced. Pollen grains are the male amplified. The pollen grains are the microsporangium is normal, where the microsporogens take place. So now, when you take the pistil is either means made up of only one pistil, or the gynoecium is made up of many pistil. If they are free, they are the apocarpus. If they are fused, syncarpus. Yes. So this is a pistil. So it is made up of stigma, style, and ori. Already you know. Stigma is a platform with the pollen grain gland. Style is the connection between stigma and the ori. Ori is the lower basal portion. So inside placenta is there. On this placenta, ovule or the megasporangium is attached. So this is the megasporangium or the ovule. Now same thing. How this megasporangium develops means, here also same archisporial cells are there. That archisporial cell divided to produce the primary parietal cell and primary sporogenous cell. Primary parietal cell divide and produce the almost the outer integument one. The sporogenous cell produce the megaspore mother cell. Yes, the same what you have seen in the male reproductive part. Same 3 cells. Archisporial cell, primary parietal cell, and primary sporogenous cell. Primary parietal cell produces the outer integument. Yes. Sporogenous tissue produces the inner gaptophyte. Clear? Now, if you take the main one, how the ovule or the embryo sac develops. So this is a megaspore mother cell. This is a megaspore mother cell. This is also diploid, which is 2n in nature. This undergo meiosis 1, meiosis 2. Yes, meiosis 2. So ultimately 4 megaspores are produced. This is also megaspore tetra. 4 megaspores are there. But out of this 4, 3 megaspore toward the chalaza, means micropylarend. This is the micropylarend and this is the chalaza. We already covered this one. So micropylarend megaspores degenerate and the chalaza became functional. Yes. if this chalazaland megaspore, functional megaspore responsible for the development of the embryo sac 1, then this phenomenon is called a monosporic development. Yes, if instead of that 2 megaspores are functional and 2 megaspores are degenerate one, if these 2 megaspores are responsible for the development of the embryo sac, then it is called the bispore. Two megaspore involved. So in some cases, all the 4 megaspores are involved in the development of the embryo sac. 4 megaspores involved. Then it is a tetraspore. So, but in your textbook, they have not only monosporic, right? 1 functional megaspore and 3 megaspore degenerate into the micropylar. This functional megaspore undergo first, second, third mitotic division. So all 3 are the mitotic division. So ultimately, if it is a monosporic embryo sac, 1 meiosis, 3 mitosis take place. If it is a bisporic, 1 meiosis, only 2 mitosis take place. If it is a tetrasporic embryo sac, 1 meiosis and only 1 mitosis take place. So based on the how much megaspores involved, mitosis is decided. Yes. So you move mitosis agute. This simple figure agute. This is a monosporic. ಇದು 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಆಗಬೇಕು. 1 ನ್ಯೂಕ್ಲಿಯಸ್ ಇಲ್ಲಿ 8 ಆಗಬೇಕಿದ್ರೆ 3 ಡಿವಿಷನ್ ಆಗಲೇಬೇಕು. ಇಫ್ ಇಟ್ ಈಸ್ ಎ ಬಯೋಸ್ಪೋರಿಕ್ 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಆಗಬೇಕಿದ್ರೆ 2 ಡಿವಿಷನ್, 1 ಡಿವಿಷನ್ ಆಗ್ತದೆ 4 ಆಗುತ್ತೆ, 2 ಡಿವಿಷನ್ ಆಗ್ತದೆ 8 ಆಗುತ್ತೆ. ಇಫ್ ಇಟ್ ಈಸ್ ಎ ಟೆಟ್ರಾಸ್ಪೋರಿಕ್ 4 ಸ್ಪೋರ್ ಇರ್ತದೆ, ಬರೀ 1 ಡಿವಿಷನ್ ಆಗ್ತದೆ, 8 ನ್ಯೂಕ್ಲಿಯೇಟ್ ಎಂಬ್ರಿಯೋ ಸ್ಯಾಕ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ. Is this clear? So number of division based question may arises. So this is about the division. So the formation of the megaspore in the embryo sac develop ಆಯಿತು. So these are the 3 mitotic division. 3 division ಆದಮೇಲೆ 2 ಪೋಲಲ್ಲಿ 8 ನ್ಯೂಕ್ಲಿಯಸ್ ಇರುತ್ತೆ. So here 4 nucleus, here 4 nucleus. Out of that one nucleus will migrate toward the center. Red color migrate here. And 3 nucleus are toward the pole. Now the cell wall is laid down. So in the mitotic division, free nuclear division period, cell wall laid down there. So now the cell wall is laid down in such a way that 3 cells are present toward the chalazaland is called the antipodal cell. 3 cells present toward the micropylar end is called as the egg apparatus. In that one, 2 synergid and 1 egg is there. So E we are to synergid to E go to egg. And central large cell is there where the 2 polar nucleus is there. So this is the 7-celled 8 nucleus structure. ಈಗ ಎಕ್ಸಾಮ್ನಲ್ಲಿ ಫಿಗರ್ ಏನಾಗುತ್ತೆ, ಡ್ರಾ ಫಿಗರ್ ಅಂತಿದೆ, ಕೆಲವರು ಹೀಗೆ ತಂದುಬಿಡ್ತಾರೆ. ಸೊ ಎರರ್. ಎಸ್, ದಿಸ್ ಇಸ್ ಅ ರಾಂಗ್ ಒನ್. ಏನು ಮಾಡಬಾರ್ದು ಹೇಳ್ತಾರೆ. The antipodal cells are present toward the micropylar end and egg apparatus is present to micropylar end. It is chalazaland. So ovule hige irodilla. This is the wrong figure. So figure ತೆಗೆಯೋದಕ್ಕೆ just draw the figure properly. So the embryo sac. So this is the embryo sac. You have to draw micropyle toward this side, E end. So this is one integument. This is another integument. This is an outer integument, and this is an anatoposole. Is this clear? So always antipodal cells always come towards the chalazal end. Egg apparatus always come towards the micropylar end. Is this clear? So this is the figure given in your textbook. So whether this type only ovules are present in our family or not. So ovulally ಮತ್ತು ಬೇರೆ ವೆರೈಟಿ ಇದೆ, ಮೆಗಾಸ್ಪೊರೇಂಜಿಯಮ್ದಲ್ಲಿ ಬೇರೆ ಬೇರೆ ವೆರೈಟಿ ಇದೆ. ಸೊ ಆಲ್ಮೋಸ್ಟ್ ಫಿಗರ್ ಇಸ್ ಡ್ರಾನ್ ಹಿಯರ್. ಸೊ ಟ್ರೈ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ ಫಿಗರ್ಸ್. ಬರೀ ಹೆಡ್ಡಿಂಗು ಹೆಸರು. ಫಸ್ಟ್ ಒನ್, ಟ್ರೈ ಟು ಡಿಫ್ರೆನ್ಶಿಯೇಟ್. ಇನ್ ದಿಸ್ ಕೇಸ್ ದಿಸ್ ಇಸ್ ಅ micropylar end, this is the chalazal end, and this is the funicle. All are in straight line only. Yes. So this is a orthotropous. Orthotropous ovule. So in this case, micropyle, funicle and chalaza all are in straight line. Curvature is not same. Example, piperacy and polygons. Yes, this is simple. ಈಗ ಈ ಕಡೆ ಗ್ರೋತ್ ಆಗುತ್ತೆ, ಒಂದು ಕಡೆ ಗ್ರೋತ್ ಆಗ್ಬಿಟ್ಟು ಸಮ್ ಟಿಲ್ಟ್ ಆಗುತ್ತೆ. Yes, so tilt ಆಗುತ್ತೆ. This is micropylar end, this is hilum and this is a funicle and this is chalaza. Yes, so funicle point change ಆಯ್ತಿದೆ. So this is a chalaza end and this is a funicle. So this is in almost straight line, means 90 degree. So this is a hemitropous. Example, malfigiacy. So ಇಲ್ಲಿ straight line 180 degree. So this is a 90 degree. Yes, hemitropous. So this is ovule where the curvature occurred. So it's sometime in E region. Almost micropyle is near to the funicle, chalaza is opposite side. ಇಲ್ಲಿ almost in 40 degree ನೋಡಬಹುದು. So this is, so only this integument is curved one, embryo sac is still straight one. So this is campylotropous. Seen leguminous. Yes, so orthotropous, hemitropous, campylotropous. Still three are there. This one, this is hemitropous, amphitropous. So ಇಲ್ಲಿ ನೋಡಿ, amphitropous. So here also curvature is there, but embryo sac is little bit curved. In the red color dot illa yadadandu embryo sac in curve agide. Here the embryo sac is a horse shoe shape. So embryo sac curve agida karana this is a amphitropous. Example, alisma. So question ide based when they are in straight line orthotropous, the micropyle funicle in 90 degree, this is a hemitropous. So where the almost 45 degree is a campylotropous. Idhar bagge maatadam. Amphitropous means where the embryo sac is also curved and it is a horseshoe shape. So next one is a typical, that is the same one. So this is the name given by the, this is called as anatropous. Question number, draw the neat label diagram of anatropous ovule under E type. Monocot and polypetalate show this type of ovule. Next one is little bit complex one. In this case, growth is there. So again funicle is the tirig mat micropylar region. This is completely 360 degree rotated one. So this is a sarsinotropous. Clear? So this is seen in the opposition. So based on this one question will arise. So bari figure ಕೊಟ್ಟು heading ನಿಮಗೆ ಕೇಳ್ತಾರೆ ಅಥವಾ example ಕೇಳಬಹುದು. So this is the most important one. Based on this one many question will arise. Orthotropous and hemitropous, campylotropous, amphitropous, anatropous, sarsinotropous. Difference ಅಂದ್ರೆ ಇದು ಎಲ್ಲ straight line. ಇದು 90 degree. This is less than 90 degree means curvature very integument ಆಗುತ್ತೆ. ಇದು 45 degree. Almost funicle comes toward micropyle comes toward the funicle only. But embryo sac is also curved one. This is a horseshoe shape. So this complete curvature takes place. Funicle is on the micropyle is near to the funicle one. Chalaza is opposite. So ಇಲ್ಲಿ so ಎರಡು ಸ್ಟೇಟ್ ಬರುತ್ತೆ. Chalaza is opposite. Micropyle opposite. Okay. Sarsinotropous means 360 degree rotation. So this is about the female reproductive part. So in next class we are going to cover the pollination topic. So as usual the PPT presentation. Yes. So Deepavali Habbada ella Anandam anniversary. So once again wish you happy Deepavali. So again we will meet tomorrow in the next video. So till then take care, goodbye, have a nice day.